Welcome to the Plant Breeding and Genomics Community of Practice webinar, the development of SNP-based Tetraploid maps for potato. My name is Heather Merck, and I'm the content coordinator for PBG and your host today. For those of you unfamiliar with the Plant Breeding and Genomics Community of Practice, aka PBG, I invite you to explore our training resources at www.extension.org/plant_breeding_genomics. I also invite you to subscribe to our newsletter, PBG News, to stay up to date with PBG webinars and more. During today's webinar, Dave Douches and Joe Coombs of Michigan State University will talk to us about their recent work to develop Tetraploid maps for potato. Today's presentation is also available at www.extension.org/pages/63187. Now help me welcome today's presenters, Dave Douches and Joe Coombs. Dave Douches is a professor in crop and soil sciences at Michigan State University. He leads a breeding program that focuses on developing varieties for the potato industry that have improved processing quality as well as disease and insect resistance. Joe Coombs has been working with the potato breeding program since 1996 as an assistant breeder focused on the program's breeding and statistical analysis. I'll now turn things over to you, Dave and Joe.
Thank you, Heather and John, for hosting the PBG webinar today, and we're happy to be here. And this is a special occasion because this is the first webinar being done from our new Molecular Plant Sciences edition at Michigan State University.
And hello, I'm Joe Coombs, and we're going to go ahead and get right into talking about the development of SNP-based Tetraploid maps for potato.
Thanks, Joe. So what we want to do today is talk about some of the work that we've done to use the SolCap-based SNPs and how we're using it to develop a Tetraploid map in potato. And this is really a team project of, that involves a large number of people, including myself and Joe, with people like Kim Felcher, Dan Zarka, Alan Van Dynes, John Hamilton, Candy Hansey, and others, and Robin Buell. Sorry about that, Robin. And so the objective of today's webinar, hopefully by the time we get to the end of it today, is that we want you to be exposed to visualizing SNPs in the Genome Studio software developed by Illumina, including the 5-cluster calling that was done here in the SolCap project. Secondly, to how we're filtering the SNPs to do the Tetraploid mapping work. Thirdly, and how we're generating the map using Tetraploid map software that was developed in Scotland. And then how we're initiating the QTL analysis using both single marker ANOVAs and also TetraploidMap. As many of you know, if you come from a potato background, there have been a number of linkage maps developed in potato going back to 1988, and some of these, or most of these maps are done at the Diploid level, and I would say that's because of the simplicity of making maps at that level than the Tetraploid level. And so you can find a number of them in the literature, and we have one that was just recently accepted that's a SNP-based one by Felcher et al. Oops, slide jumped ahead there. So with SolCap, one of our goals was to develop a genome-wide set of SNP markers, and under that work, there's a number of activities that we're doing on the potato side of the project. And one is we wanted to really assess the concordance between the map location of the SNPs and the potato genome sequence location based upon the pseudomolecules that's part of the potato web browser. And so that's the Felcher et al. 2012 article that was just accepted in +1. What we also want to do and what we're focusing on today is using this Infinium 8303 potato array to genotype tetraploid populations and to generate a SNP-based genetic map. And we're also going to touch upon our initial efforts for QTL analysis in the tetraploid populations using the Tetraploid map. And we're also in the project, but not going to talk about today, is the association analysis of the potato diversity panel and that's being headed up by Candy Hansey and Robin Buell's program. The diploid map that was generated was really based upon two maps. One we refer to as DRH, which was a cross between DM, the doubled monoploid, and RH, a Dutch diploid clone that was also used in the genome sequencing. D84 is a cross between DM and 84 SD22, a diploid from our breeding program that seemed to have a high level of SNP polymorphism. Just jumping to the results of this is that we were able to, with this SNP mapping at the diploid level, to show that the SNPs were distributed well throughout the chromosomes and across the genome, as you can see here in chromosomes 1 to 6 and 7 to 12. If you look at it from a table perspective, what we have here is, you can see, I'll use my cursor here, that in the column under DRH, you can see the number of SNPs that we were able to map as 1,960, and in the D84 we were able to map 2,454. With 787 common, we were able to map 3,627 different SNPs back to the, and compare that to the genome browser position. If we jump over to the right side of the table here, you can see the estimated map size for the two different populations. And it's interesting that we have about a 30% reduction in the population size in D84 from DRH. However, when we look at it from a pseudomolecule perspective, that the coverage across all the pseudomolecule, the 12 pseudomolecules, is that we have 634 and 642 megabases covered with our SNPs, and we know that there's about 650 megabases in the 12 pseudomolecules. So we felt that that work really provides a foundation for us to move forward, now knowing that the map position for most of the, or a lot of the SNPs that we're working with. And so we jumped into our tetraploid mapping work, and one population that we're working with that was a key to the SOLCAP project is one that's a cross between Premier russet and Rio Grande russet, and it was a cross made by Rich Novi and in the USDA ARS organization in Idaho. And this population has been grown in 3 locations over 2 years for us to collect phenotypic data, and then we've also done the SNP genotyping here at Michigan State University in our SNP genotyping lab. And so I have to give thanks to Rich, Novi, and Craig Yencho, and Christian Thill for collecting the phenotypic data, which will be part of the QTL analysis. The traits that were collected in this population is shown here, but the top 3, specific gravity, chip color, and glucose and sucrose, were some of the key important traits. And the data was replicated twice, and we used 10 plant plots as our experimental design. Today we're going to focus on, when we're looking at the QTL analysis, on the specific gravity data that was collected. For people that are not familiar with potatoes, specific gravity is an estimate of the potato tuber's density, which is has a good correlation with the starch content of the potatoes. So a higher starch content is desirable for the processing of French fries and potato chips. This russet mapping population is part of a broader set of germplasm that we've looked at in SOLCAP. Also in this broader population is our diploid mapping populations, which I've talked about already, as well as our diversity panel, which I mentioned earlier. And in this diversity panel, we have 325 clones that we've SNP genotyped, and of those 325, we've collected phenotypic data on about 220 of those lines over 2 years at multiple locations in the U.S. So that's going to be part of some future analyses. There's a number of other populations that we've SNP genotyped under the SOLCAP umbrella, and these are listed here. The first 5 populations are tetraploid populations, and the one at the bottom of the list there is a diploid population. And you can see the traits that we're measuring or in these different populations, and it'll be very interesting as we start get through the QTL analyses and being able to validate certain QTLs for similar traits across the different mapping populations. Just for people that are not familiar with the SNP development as part of the SOLCAP project, our SNPs were developed from elite germplasm, so we used 3 varieties, Snowden Atlantic and Premier Russet. And from those lines we collected RNA from 4 different tissue types, as you see here, tuber, leaf, flower, and callus. And from there we were able to then get a good broad look at the transcriptomes of these 3 varieties. We used Illumina GA2 sequencing. Alan Van Dynes did the work at UC Davis and generated approximately 2 gigabases of sequence information on each of the lines, which gave us about 50 to 60x coverage of the transcriptome. Robin Buell's lab, with a lot of work from John Hamilton, did the bioinformatics work and sorted through all the SNPs, and we had over 500,000 different SNPs that we could consider for using. But to fit the Illumina Infinium platform design, we had to go through a certain specific filtering steps. And when it was all done, we had 69,011 SNPs that passed all the filtering steps. And all these SNPs can be found on the Potato Genome browser. So we chose to make a 10K Infinium array for the potato, and when it was all said and done, we had about 8,000. Well, we had actually exactly 8,303 SNPs that were in this SNP array for potato. And if we look at what made up these SNPs in this final number, was about 3,000 of them were from candidate genes that were suggested by the community. So we found all the SNPs that we could in those. Plus, we found some SNPs that were in previously mapped genetic markers as a reference point. Plus, the remainder of the SNPs coming from ones chosen throughout the genome to maximize our genome coverage. The Infinium, Illumina Infinium system is based upon single base extension, and so just to kind of walk you through that platform design of our potato array is that what you have is a large number of beads, and on each of these beads is an oligo specific to the SNP that we're trying to identify. And Illumina creates a bead pool of about 250,000 beads per sample. And that sample over here is one of these 24 wells on this chip that you see over here. And then what goes through is a random self-assembly of these beads onto the chip. You can see them going into the wells over here. And then when you average this all out, there's a redundancy built into the system. So for each SNP that we're trying to read, there's about a 15 to 30 bead redundancy of that SNP. And so when we do get a call, it's really based on the average of those calls for that SNP. And so we have the 8,303 SNPs on the chip, and on each chip we can have 24 samples. It's about a 3-day operation to do the SNP analysis if we have good quality DNA to start with on the first day. And when the process is done, the bead chips are loaded into the Illumina iScan, and then a laser is used to excite the red and green fluorophore, and that then is read into the computer software, and it records this high-resolution image, and you can see this over here, and you're getting the red and green emission of the light from the different beads. So this data then is matched up with the mapping software that comes from Genome Studio of where each SNP is on the chip, and then that's imported into our Illumina Genome Studio software. So you have to have this software to work with the Infinium chip, and just for people that aren't familiar with it, what we're looking at here is a screenshot from that, and I have the cursor along one of the rows, rows of our SNPs. So this column here is our various SNPs, and we have them sorted in some order. But the key values that I want to point out here is the theta value and then the R value here, and those two values are used to graph the SNPs onto this graph here, and over here, which you can see is actually the progeny, or the samples are listed, and we can kind of color highlight different samples within this graph for ease in viewing. So now the Illumina system the Illumina genome software has auto calling for the SNPs. However, in the SOLCAP project, what we did was we actually created a custom potato calling, and for the 3 clusters, of the 2 homozygous classes and the heterozygous class, and we based this upon our potato diversity panel, 2 tetraploid populations, and 1 diploid population. And so this custom calling file can be accessed at our SOLCAP website. So of the 8,303, it was determined that 7,412 of those SNPs were considered good for 3-cluster calling. So if we're looking now closer at those Illumina Infinium graphs of our SNPs, what we're looking at here is one SNP example, and what we have here is one parent in the homozygous AA state, and we have one parent in the homozygous BB state, and the progeny from that should be expected to be all heterozygous AB, and that's what we see here. And on the second graph in this slide, you can see we have one parent is a heterozygote AB and the other one's a homozygote BB, and when we cross them, we have a 1-to-1 distribution in the progeny for the heterozygote and homozygous classes. So it's a very easy system to read, and what we're seeing along the x-axis here is the theta values. So where the theta value is kind of helps us position the call on the SNP, and the R value I think is a reflection of the intensity of the signal that the system was reading. Okay, so we're looking at it now we've looked at it from a diploid level. So what we really need to do is start thinking about tetraploids, and so that can get some people's heads swirling, and or lead to some nightmares. But when you have tetraploids, you have a different type of segregation patterns than what we normally see at the diploids, and so we can have 1-to-1 segregations, 1-to-4-to-1 segregations, 1-to-2-to-1, 1-to-5 to 5-to-1, and then the most complex is the 1-to-8 to 18 to 8-to-1. Okay, so what's happening in each of these tetraploid segregations, we have up to 5 different genotypic classes, and so the Illumina Genome Studio software is designed for calling 3 clusters, and what we need to be able to do is call 5 clusters so that we can differentiate between the 3 heterozygous groups, which we refer to as simplex, duplex, and nulliplex, which are the different doses, in this case, of the B allele, of a single dose, double dose, and a triple dose. What's really been exciting about this Infinium platform is that the SNPs are codominant, and many of them are dosage sensitive, and so we really do have a very ideal marker to look at our tetraploid populations. So as a next step, the same panel of potato lines, the diversity panel, the 2 tetraploid populations and the 1 diploid population, were then looked at to develop a 5-cluster custom calling based upon the theta values. So each SNP was looked at, and there was a lot of effort put into this, and as a result of that, it was determined based upon our data set that we have a little over 5,000 SNPs that we can use the 5-cluster custom calling. And we had about 3,272 that there was only 1 cluster or was a bad SNP. So now we're looking at our tetraploid populations here on the Illumina Genome Studio graph, and we have here, we can see we have parent 1, parent 2. On the left side we have a diploid population, and again we have the heterozygotes all clustering to the middle of the graph. On the right hand side we have a tetraploid population. We have a cross between a duplex heterozygote and another homozygous parent. Oh no, sorry, take that back. We have a cross between our simplex heterozygote and our duplex, and so you can see then the segregation between the 2 genotype classes there. And so we consider this ideal because it works both for the diploid and the tetraploid germplasm. So now what we're going to do is look at some of the data that actually came off our premier russet by Rio Grande russet mapping population. So as you saw earlier, there was 184 progeny that we're basing this mapping population on. That number is the number of progeny where we have both phenotype data and genotype data in a usable form, and to do the analysis. So looking at this first graph here on the left-hand side, what we have is a cross between premier russet being a simplex heterozygote to Rio Grande russet being a nulliplex. And so what we expect is a 1-to-1 segregation between the nulliplex and simplex classes. And you can see the data up here, and we thought it was quite cute that the numbers came out exactly. We are not trying to be like Gregor Mendel and make our numbers fit, just for people that are questioning things. And oops, sorry, I jumped ahead by accident. And then on the right-hand side, we now have a cross between our nulliplex for premier russet and a duplex SNP in Rio Grande. And here we expect a 1-to-4-to-1 segregation ratio, and here you can see the numbers that we obtained in our progeny, and those fit that expected ratio. Now just one thing to point out here is on our screen here we're seeing the circles drawn for the auto-calling in Genome Studio, whereas what we're doing is actually basing our SNP calls on the 5-cluster custom calling. So those circles are kind of something that's difficult to remove from the graph at this point. Okay, so now looking at a different segregation pattern, what we have here is a duplex heterozygote crossed to a simplex heterozygote. And that's going to give us that 1-to-5 to 5-to-1 segregation ratio. So we have 4 segregation classes that we're scoring in this, or 4 genotypes that we're scoring in this progeny. And so you can see again we've got the separation into those our heterozygotes into the 3 heterozygous genotypes. And then lastly, what we have is that complex duplex by duplex segregation, which gives us the 5 genotypic classes and segregating in that 1 to 8 to 18 to 8 to 1. Now, as we were looking at the data, we kind of faced a reality point. And one of the questions we always had is, well, how good is the SNP calling? You know, are things going to be miscalled or not? And when we started looking at the data, so in a simplex, nullplex by simplex, as I showed earlier, we expect the 1-to-1 segregation. However, because potatoes are tetraploids and have autotetraploid type segregation, one of the consequences of that is we can actually see products of double reduction. And hopefully my potato colleagues are familiar with this term. And so this brought back some fond memories of my earlier days of looking at double reduction. And so what we are seeing double reduction products with the SNPs. So what we have here is we expect the 1-to-1 segregation between for the nullplex and simplex, but a double reduction product in this cross would be a duplex, and we can see them clustering separate from the simplex. And we have on the right-hand side we have another example of that in a cross in the other direction where we have a B homozygote to a B to a ABBB simplex or triplex. So so we were seeing this a number of times. And so if you go back to your cytogenetics, what you can see is that the double reduction is going to happen when you have autotetraploid pairing in meiosis. And if you're really interested in this subject, you can go to this book by Burnham, Discussions in Cytogenetics, 1962. But just as for you to walk you through this, is that double reduction occurs when the 2 sister chromatids are recovered in a single gamete. And what this requires is multivalent pairing, and then a crossover between the locus and the centromere, and then the 2 pairs of the chromatids passing to the same pole. And so if you were to work out the probabilities of this, it happens about that in a simplex cross, we're going to see that approximately as a maximum of 1/24 of the time. So, but it's the frequency of the double reduction is proportional to how far it is from the centromere. So loci that are closer to the centromere, we're not going to see that. And so this table here, we graphed out all 800 simplex segregation patterns that we saw in our SNPs in this population. And so 541 of the SNPs, we did not see any double reduction products. And as you can go down the list, you can see as the number of double reduction products goes up, the number of SNPs that had those number of double reductions goes down, because the SNPs would have to be far from the centromere for us to see those. Okay. Using some graphical software in the statistical program JUMP, Joe Coombs was able to plot this out. So what we're looking at here is we've got our 12 chromosomes, 1, 2, 3, and so on. And what we have across is the linear order of our chromosomes in megabases. Okay, so what we'd expect to see, and I think it's seen clearly here on chromosome 4, is that our double reduction products would tend to be more frequent at the ends of the chromosomes where they're far from the centromere. So you see this clearly in chromosome 4 and in chromosome 5 and 6 and so on. This we haven't really delved into this data much, but this is what we're seeing so far, and so I just wanted to pass that on, because double reduction is something that's happening, and we got to be aware of it. And if you're using the TetraploidMap software, it actually does not handle double reduction products, so those have to be removed from the analysis. Okay, so getting back to the presentation here or getting back on track. So what we need to do is filter the SNP data. And so we had at the start 8,303 SNPs to start with on the Infinium SNP array. And I have colored in yellow here, shaded in yellow, the SNPs or the filtering process that is really general to anything we really do. We need to be able to remove the questionable and bad SNPs, ones that aren't anchored to the pseudomolecule, ones that map to more than 2 locations. That's kind of standard. And whether it's a diploid or tetraploid population. But as I said earlier, there was only so many SNPs that we felt we could use from the custom 5 cluster calling, and that was a little over 5,000. So when we did this initial general filtering and then looked at the custom 5 cluster calling, that filtered it down to 4,604. We've used 10% as a cutoff for no calls in the progeny. So if we have more than 19 progeny where we did not get a call on a SNP, that was removed from the analysis. So that took out about 400 different SNPs. If the parents had a no call, we removed those. That didn't remove too many. And then we removed any ones where there really wasn't any segregation in the progeny. So that gave us actually 3,298 segregating SNPs in this Premier Russet by Rio Grande Russet mapping population. This table here is showing us the frequency of the different SNP combinations that we have in this population. So on the left-hand side, you can see the Premier Russet genotypes, and on the top, we can see the Rio Grande genotypes. And then what we have is shaded in color the nulliplex by simplex, and that adds up to 800 markers that we can use. Now again, this is kind of interesting, is that the TetraploidMap software has a maximum of 800 SNPs, and so again, we are not manipulating things. We just have an interesting way the numbers are sorting out. Okay, so now we can also use nulliplex by duplex in the mapping, and so there ends up being 371 of those SNPs segregating in that manner in this population. And then lastly, you know, a nulliplex by triplex is really like a nulliplex by simplex cross, just in the other direction, and we have 38 of those. So that's our initial set of markers that we can use for analysis. These other ones are segregating, you know, that we have the duplex by duplex or duplex by simplex, but we're not going to focus on those yet in the population. And so one of the things we can also do is, after we see which SNPs we have, is, well, where are these SNPs distributed along the chromosome? And so again, Joe was using the JUMP graphical interface to look at that. And so what we have here is, in red, the whole pseudomolecule for each of the 12 designated chromosomes in the potato. And the red marks are the SNPs, where the SNPs are mapped on those. In the green, what we have is the Premier Russet parent, and you can see where the SNPs are. And in the blue, we have the Rio Grande Russet parent, and you can see the distribution of the SNPs. So in general, we had pretty good coverage across the genome. However, there were some gaps, like here on chromosome 3, and also on chromosome 9. And then even within some of the chromosomes, we had some gaps, like here on chromosome 12 and also on chromosome 11. So there, but this is a nice way to visualize how we're seeing things. And this table here summarizes those numbers for you. And we left this in the webinar for people that view it at a different time, so they can just have the numbers rather than just a visualization. So I'm not going to focus on this at this point. So now we're getting to the point of doing the QTL analysis and some of the mapping work. Okay, and so what we wanted to do simply was, let's take a trait, and we took what we considered a quantitative trait, specific gravity. We took one set of data. We took the 2010 Idaho data. We give kudos to Rich Novi for that. We figured that he had a good data set for us to work with, and we said, let's just do a single marker ANOVAs on all 800 simplex segregating SNPs. And so you can see in the Premier Russet we have 487 SNPs, simplex SNPs segregating, and Rio Grande we have 313. So of those, 56 and 77 SNPs ended up having significant probabilities for or significance for specific gravity. If we were to use the multiple test corrections, or the Bonferroni or the Sidak-Dunnett, it actually none of the SNPs actually show significance. However, let's just take a moment and look at this. Now again, what we have are our 12 chromosomes, and what we have along here is the length of the pseudomolecule. So it's going from 0 to 80 megabases for chromosome 1. And what we have is the dots mark each of the SNPs that we looked at in Premier Russet for the specific gravity significance. And this red line drawn here along the table, the figure, is our cutoff for the 5%. So if those dots are below the line, they were showing the statistical significance in the single marker ANOVAs. Just focusing in on that chromosome 1 at this point, and you can see that we had 3 SNPs that fell within that 0.05% range for significance, whereas the others didn't. So let's just kind of keep that there. Just to make a point, you can see that we had actually 46 SNPs in this along this chromosome arm, and also when we're using the map TetraploidMap software, it only will take a maximum of 50 markers per chromosome at a time. So it was nice that our data was falling within those limits of the software. Here's another way using the JUMP software that we could visualize the data from the single marker ANOVAs for the simplex markers for specific gravity. So what we have here are the 6 chromosomes where we found significant ANOVAs for those for specific gravity. Now, and again, these are in linear order along the chromosome. So we have these color-coded. So if it's homozygous for either the A or the B, they were color-coded blue. If they were heterozygous for either for AB, then they were color-coded red. So what you can see here is for each SNP, was it the heterozygous condition red or the homozygous condition blue that was giving us the higher specific gravity or the lower specific gravity. So that's just a nice way to visualize data. And so you can see that we had multiple SNPs at times, probably identifying a similar QTL on the chromosome. And here we had a very large cluster of SNPs that were in there. So you can see that. So we have 6 chromosomes with showing significant SNPs for specific gravity using the single marker ANOVAs. That was in the Premier Russet. Now we're looking at the Rio Grande. And again, instead of the 6 chromosomes, we have 5 chromosomes showing SNPs that were significant for specific gravity, and they were common with the Premier Russet. So I thought that was some good validation of what was of potential QTLs to start with. And again, we have the same color coding where the blue is the homozygous condition and the red is the heterozygote. So you can see here that we had a number of homozygotes that were given the higher specific gravity. So now we're getting ready for using the TetraploidMap software, and we have the link here from where you can download this. It's you need to use Windows XP software at this time, and it was originally designed to be used with AFLPs and SSR markers. And as I mentioned earlier, that there's the maximum right now is 800 markers, but Christine Hackett's expanding this so that it can handle a larger number. And on a per chromosome basis or linkage group, you only can use a maximum of 50. And they also state in the software it's not effective for markers with double reduction. So we have to remove that. Now, this is where I feel that having the potato genome sequence prior to this mapping work has helped us. By doing the diploid mapping work, we were able to see the concordance between the SNPs in the diploid population and then the potato browser. So we can use the position of the SNPs on the pseudomolecules as a way of organizing our data in TetraploidMap for doing our analyses. Without that, we would be having to do things on a random basis, which would really slow the analysis process down. So Joe Coombs has been the person that's been diving into this software and handling all of the data for the Tetraploid map. And so Joe's going to take over here and kind of walk you through some of the work that he's done in the Tetraploid map.
Thank you, Dave. As Dave was saying, TetraploidMap is probably one of the only, or widely available, freely available Tetraploid mapping software programs out there. And this was really our first work using that software and incorporating the SNP markers in the program. And we wanted to just give a little bit more background on how we worked with our data to bring that into TetraploidMap and what we were seeing with some of the initial results. With working with the SNP markers, we began by recoding the 5-cluster calling so that it would go into the numbering format for the TetraploidMap software. And this table is just an example. We're working with the data originally out of Excel, but this is a list of the SNP names, and since we have the pseudomolecule information, we had already incorporated the pseudomolecule chromosome and position, and then you can see the 2 parents, Premier Russet and Rio Grande, and then a sample of the progeny. So when we were recoding, first we were looking at those 800 simplex by nulliplex SNPs. And so we would recode an AAAB or an AAA as a 1 and a 0, as basically coding it as a dominant marker for a TetraploidMap. And then when you do have missing calls, they're recoded as a 9. So we were doing that with no calls or with calls that were double reduction products. When we're bringing in the marker data, it has some formatting requirements, and so the data is laid out. So you begin with the number of progeny that are being put into the project, and in this instance we had 184, and that does not include the 2 parents. So there are a total of 186 genotype calls that were being incorporated. And then the number of markers that are being put into the project. And so here you can see there are 46, and this was looking at 1 parent, the Premier Russet, for a single chromosome, chromosome 1. And we had broken these out to work at a single chromosome parent at a time in TetraploidMap, so that when we got to the marker ordering stage, we would be under the limit for the ordering numbers. Next, you just have the marker name here, and then this is the number of alleles to expect, and in this case it was 1, and then these are the actual genotypic calls for the parent 1, parent 2, and then the progeny. So The TetraploidMap software is actually a very inclusive software package, and once you incorporate your marker data, you can use the software for cluster analysis and following that marker ordering, and then also QTL analysis. And I didn't want to go into a lot of detail, but just to kind of direct people to the webinar that is currently already available online. Christine Hackett is one of the developers of the software, and she had put on a very nice workshop in the SOLCAP workshop for the Potato Association of America meetings in 2010, and the link is here for people to go and get more in-depth background on using the TetraploidMap software. So after we incorporated the markers from first parent premier russet chromosome 1, we were able to do marker ordering, and we had the overall marker order that had come out of TetraploidMap software. And then just as a reference point, we wanted to see how the genetic map positions compared with the pseudomolecule. And this is an example of chromosome 1 for premier russet from the tetraploid cross. And on the left side here, we're looking at the genetic position that came out of TetraploidMap compared to the physical position on the pseudomolecule. And you can see there's a nice correlation there. And then referencing one of the two diploid populations, we were also seeing the same thing from the diploid map, where we're looking at the genetic position from the DM by RH population compared to the physical position. So it was nice to see a good correlation for that marker set coming out of TetraploidMap. So after we had the marker ordering, we were moving forward to do the QTL analysis, and there's also a little bit of formatting of the data that needs to be done for the phenotypic data that goes into TetraploidMap. In this instance, we start with the number of traits that were being entered. We had 21 different traits, and then it's just a list of the progeny, and this corresponds, of course, to the same progeny order that were put in for the genotypes, and then the different trait names and the data for those traits. And in this instance, any missing data is coded as a -99. In order to do the QTL analysis, you need to call the chromosome for each one of the markers individually. And there's more information again from the workshop that Christine Hackett had given, but basically working through each individual SNP at a time, looking at whether the SNP is in coupling or repulsion, and determine the chromosome for the parent that it's on. And so in this example, these first three markers were arbitrarily assigned chromosome 1, and then this next one was found to be linked in repulsion, so we put that on chromosome 2, and so on until you're able to call the different chromosomes for each one of those SNP markers. So after doing that, we're able to see not only the overall position on for chromosome 1, but then actually each one of the 4 different chromosomes in that parent. So we will have a map not only for each parent and chromosome, but the actual 4 different chromosomes of each one of those parents. So after calling the different chromosomes, you can move on to doing the QTL analysis. And this is just an example, again, looking at the specific gravity. And in chromosome 1 for premier russet, you're able to see in that similar region where we are seeing some significance in the single marker ANOVA, it's nice to see that we're also seeing a good LOD score for the QTL analysis in that chromosome. So I'll pass it back to Dave to wrap up.
Yeah. Well, thanks, Joe. What's really something to think about when you're doing the tetraploids is that when you cross two heterozygous tetraploids, you're actually developing two different maps, one for each of the parents. And so we were doing the analysis by chromosome. So there's actually 24 separate parent by chromosome analyses that need to be done for each trait. And but then within each chromosome, you have then those 4 chromosome maps that are made for each chromosome because of the way the coupling and repulsion works in the tetraploids. So you're really ending up with a lot of different maps in a tetraploid potato, as some of our colleagues would know already. So what we want to do is just summarize. We went through the 5 cluster calling. We showed the steps of filtering that we were using in our tetraploid population. We demonstrated how we were using the TetraploidMap software for chromosome 1 in Premier Russet, and how we initiated some QTL analyses with our Simplex markers. And so one of the goals of SOLCAP is to develop and maintain databases and resources for the tomato and potato community. And so these databases can be accessed here on this slide, and as we move through our analyses and all that, data will be uploaded to these different websites. And also we feel that one of our goals is really to contribute to this Breeder's Toolbox, which is part of SGN, with the idea of making a set of data useful to the breeders that can be found within SGN and accessed by the breeding community. And SOLCAP is kind of in the later stages of its project, and in 2012, which is what's going on, is that we're going to be continuing to do the SNP genotyping on the panels and populations, or that's been done, and now we're working on getting the database uploaded with the genotype and phenotype data, and then mining this data, and also continuing with the QTL analysis on the different populations. And we're planning to have some type of hands-on workshop for the breeders now that we're developing some expertise in this area. And also this eextension.org is an important arm of SOLCAP, that's for us to have these webinars and other information that you can access through the PRRG site. And then lastly, just need to acknowledge all the co-PIs of SOLCAP, Dave Francis and his lab, Walter DeYoung, Lucas Mueller, and their group there at Cornell, Alex Stone and her group there at Oregon State, and at Michigan State, the group with Robin and I, and Alan Van Dynes at UC Davis. So at this time, thank you for listening in, and for questions. Heather?
Thanks very much, Dave and Joe. That was a really nice talk you guys gave. So we have about 10 minutes left today for questions. So I want to ask you, unfortunately at this time we're not able to answer questions, we're not able to take verbal questions. So if you have any questions, comments, please type them into the chat box. And if you missed the beginning of the presentation, you can just type your question in the question box on your screen and hit return. If the question box is closed, please click the small plus sign next to the word question to open it up. And I'll be reading the questions out loud for Dave and Joe, and we will answer as many as we have time for today. And so there was one question that was asked early on about the how the SNPs are detected, and I think that it seemed like later on in the presentation you guys went through that pretty much in detail. So I would ask the person who did pose that question, please type me a little message if your question wasn't answered adequately. And so we've got one question here. The question is, were you including the nulliplex, the duplex by nulliplex markers in the QTL analysis?
Not at this point yet. We've added the simplex in, and our next step is to now add back in the duplex. Because of the limitations of the software in terms of how many markers it could handle, that was kind of putting us over the limit on the total and also by chromosome. So that's kind of our next step.
Great. And we've got another question here. Hopefully, I think, Joe, I know you've talked some about this, asking how you make the 5-cluster column file, doing this in a manual way, SNP by SNP.
So the 3-cluster calling file has been released for Genome Studio and is available from the SolCap website. And right now the 5-cluster calling file that was actually done by Candy Hansey and Robin Buell's program. And at this point it's just at the stage where it's a Perl script that works with the theta values individually from those files. And so I'm not sure at what point in the development if that is going to be a custom file in Genome Studio, but I think that that's the direction that it's moving.
It'll be, I think, made publicly available when we get our publication out on the Potato Diversity panel.
Great, that sounds wonderful. It's since we've been just chatting here, there have been a number of questions coming up about the clustering file. So that sounds like that'll be a good resource for us to be able to make available in future. And another, go ahead.
The diversity panel paper manuscript is being worked on at this time, so we expect that to be, come out in a relatively short time. Not in a couple weeks, but in a couple months. Okay.
Great, thanks. And there's a comment here. So thank you very much for this person who said that there is, for those of you familiar with R, there is a 5-cluster calling, looks like package available within R, and that's called Fit Tetra. So for those of you who are familiar with R, it sounds like there are some resources available at this time that you can work your way through. And another question here about TetraploidMap. You guys have talked some about the limitations, and that Christine Hackett, her group, as we get more and more data and more resources available, that she's looking to expand. Do you have any idea if a later version might be available for Windows 7?
We actually recently spoke with Christine Hackett, and we know that they are currently working on developing a new version of TetraploidMap that's designed specifically to handle SNP marker data similar to what we're working with right now. So to handle larger data sets in terms of number of markers, and also to handle the 5-cluster calling information. But I know that they're still developing that. I'm not sure what the release time frame is on that. Currently, with Windows 7, I've heard that TetraploidMap can be run in compatibility mode, but I've still found, for me, the best way to run it is with Windows XP on a virtual machine, so something like VMware or whatever other virtual machine you might have, or an actual Windows XP machine.
Sure. Thanks very much for that, Joe. And a couple questions related to the QTL analysis here. So one question is, or comment, there seemed to be no major specific gravity QTL on chromosome 5. So the person asking the question is wondering if you corrected specific gravity for earliness.
We did not do any correction for earliness at this point. it was just based on the straight data. Okay, I'm just trying to get to my map here and see where we had the. So actually, we do have one. When I look at the data, we do have one on chromosome 5 in Premier Russet, and I guess we. Hold on. Yeah, so there's one in chromosome 5. Yeah, and they're also on chromosome 5 in Rio Grande. So,
okay.
So we do have a group of them there.
Great. Thank you very much. And another question related to the QTL analysis for specific gravity, wondering if to this point you've looked at any QTL by environment interactions.
No, right now what you're seeing is what we've been able to do. We have our we've just delved into it. We wanted to focus on the one data set. We've been trying to get the software working for us in an efficient way, have everything formatted. So now we're going to be pushing forward with the broader data sets as we move to things. Our first goal is really to get the duplex markers incorporated, and then move to the larger data sets.
Great, thank you. And there was another request here to repeat the name of the R package for 5-cluster calling, and it's called Fit Tetra. And I'll also just take a moment here and type that into the chat box. So that's an R package used for 5-cluster calling. Okay, we've got many questions here, so it's good to see we have a really active audience today. The next question is, what do you think might cause the clustering of SNP markers in homologous groups? They have some concern that there may be too many gaps for QTL interval mapping.
Oh, well, as I think I pointed out, that we did see some gaps, and I think it's when we actually look at the that chromosome 1, and you can see when you divide that into the 4 different chromosomes within your tetraploid parent, there are some significant gaps, and so that is a little bit of a, or not a little bit, that is a concern to me. And really 800 markers is not really complete coverage. We really need to expand that. So when we throw the duplex in there, that will help things out. And then if we're able to use some of these other marker segregations in the future, that will help fill in the gaps. We still also, this was based upon our 8303 array, and what we can do is also go back to the browser and maybe consider other sets of SNPs that might fill in some of these gaps that we're seeing. But I think that's a very good observation, and clearly that we haven't reached the final stage of tetraploid mapping in potatoes with this SNP chip. And this is only our first population that we're looking at it, but I expect to see gaps in the other populations too.
Mm-hmm. Absolutely. Yeah, thanks, Dave. And just as a comment here, we do very much appreciate the willingness of your group and Robin's and the other collaborators of this project for really helping us get a sneak peek and giving us a little insight here. So it's very much appreciated. And there is another question here. Is my understanding correct that you mapped QTL separately in the male and female parents? That the
Yes.
Okay.
Okay. Go ahead, finish the question.
Yep. So the question says, as I'm sure you're aware, the phenotype will be the result of both male and female allele contributions. So what is your strategy for integrating male and female meioses into a single map?
Well, that's where the duplex markers come in, and that will actually bridge the 2 maps at this point. So when we add those in, that will help, I think, synthesize 2 maps. That's what in our discussions with Christine Hackett a couple weeks ago, we had a discussion about that.
Great. And there's a question here asking if you're aware of any haplotyping software that may be appropriate for tetraploids.
I'm not familiar with that at this point.
Sure. Yep. Okay, we're running out of time. I'm trying to choose maybe just one more quick question here. So the graphs on the amount of double reduction did not show a monotonic increase towards the ends of the chromosomes. Do you think these are the results of poor ordering of the superscaffolds into pseudomolecules?
Okay, we're just looking at our slides here to get to that, to look at that a little closer. So in this initial run of looking at the double reduction by chromosome position on the pseudomolecules, as I pointed out, there were a number of them where it matched up quite well, but there are a few places where it seems like we've got a jump up of in the wrong place along the chromosome, maybe in the centromeric region. So we haven't delved into that to get a better understanding of that, but it is an interesting point that question whether we have some misorientation. But a lot of we were able to kind of correct a lot of orientation of the pseudomolecules with the diploid mapping that was done earlier. So yeah, their question is good, and it's something that we're going to try to examine closer when we look at this data. It was kind of a sidebar to this whole analysis.
Great. Well, thank you very much, Dave and Joe. As I mentioned, we are unfortunately out of time today. We had such a great discussion going. It's unfortunate to have to cut that off. And I wonder if you could just advance one more slide for me. I believe we've got the link to the presentation. Yes. So there was a question if we could provide a link for the presentation. So there's a PDF available at eextension.org. And we will be working to make the recording available hopefully within about the next week. And to wrap up, I'd just like to thank Dave and Joe very much once again. It's been a pleasure to chat with you guys today. So thank you very much.
Thank you, Heather.
Thank you.